Due to the inherent complexity of many biological systems, as well as the stringent requirements necessary for biocompatible cooling and warming, precision and speed are crucial metrics in biopreservation. However, these metrics are quite limited when materials and processes are done manually. This is where machine automation, and specifically robotics, can come into play. Robots are generally any operated machine replacing human effort. While intricate human-like machines, such as the Boston Dynamics robot, may come to mind, what is currently more applicable in the realm of biomedical engineering and biopreservation are arm-like machines dedicated to dispensing as well as movement of materials. With the robot shown here, motion and speed in the X direction, Y direction, and Z direction is controlled by a computer program. In many biological handling robots, material interfacing is handled using air pressure. A dispenser such as the EFD Ultimus 5 can modulate the amount of air pressure at its outlet. The dispenser is connected to a compressed air source at its inlet and to syringes on the robot by pneumatic tubing at its outlet. On the dispenser screen, you will notice that there are two pressure functions, PRES, or pressure, and VAC, or vacuum. The pressure function generates positive pressure, and the vacuum function generates negative pressure. If you want to push air out of the system and through the syringe on the robot, you would use the pressure setting. If you want to create suction through the syringe on the robot, you would use the vacuum setting. How does a system with robotic motion and air pressure control relate to biological handling and biopreservation? If a dispenser-connected syringe is loaded with ink and a piston is inserted, positive pressure moves the piston down, which evenly pushes the ink down the syringe. More viscous inks, such as hydrogels, create filament strands when extruded and can be used together with a motion control system to create complex shapes and objects with precision. This is a form of 3D printing called direct ink writing. Inks with lower viscosities, such as cell media and/or cryoprotectant solutions, instead create precise droplets. Importantly, at lower pressures, you can dispense ink containing cells without the pressure causing biological damage. At the University of Minnesota, we have utilized a robot system to generate precise droplets of cells in cryoprotected solution for the purpose of vitrification. As shown in this video, droplets were dispensed onto a liquid nitrogen-covered substrate. It was found that the droplet volume is dependent upon the size of the needle, input positive pressure, and the length of time the pressure is applied, or known as the pressure pulse. The droplet size varies minimally otherwise. This is important because, as shown in this next figure, cooling and warming rates inside the droplets are quite sensitive to the droplet size. Thus, the dispensing control of the robot played a crucial role in biopreservation outcomes. The same system can be used not only for dispensing materials, but also for precisely picking up and placing them. For this application, an empty syringe can be connected to the pneumatic tubing. Turning on the vacuum setting on the dispenser connected to the other end of the pneumatic tubing generates negative pressure through the syringe. This can first be used to pick up an object, as shown on the right. By direction of the computer program controlling the robot, the syringe moves to the desired XYZ coordinate for picking up an object, and at this point the dispenser activates the vacuum pressure. Depending on the environment which the object is residing in, it might be easier to move the substrate, such as a cell culture dish, by hand rather than pre-pick the exact location of an object. However, if the object is stationary on a platform, manual interference is not necessary. In addition, a vision system can also be incorporated which can track objects of interest, provide feedback to the computer program, and then specify or update the picking location. Either way, suction will attach the target object to the needle, even after it moves from the picking location. Typically, the amount of negative pressure needed to pick up materials is much lower than the positive pressure needed to dispense materials. Higher vacuum pressure could potentially deform or damage certain objects, especially those that are soft and biological that may need to be cryopreserved, such as spheroids or embryos. The next step is object placement. Again, by direction of the computer program, the syringe moves to the designated XYZ placement coordinate, and then the negative pressure is turned off, removing the suction between the needle and object. If the object is placed on a solid surface, the tip of the needle should be a length above the surface corresponding to approximately the width or diameter of the object. This way, the object is not dropped and there is minimal impact force. Then, the program will move the syringe up and away from the placed object. You could potentially write a computer program in which, after this step, The syringe moves back to pick up and place another object, and you could repeat this process any number of times for higher throughput. You could also instead place objects in a liquid or gel, which might be more useful depending on the application. We have utilized negative pressure-based pick-and-place robotics for handling and cryopreservation of organisms. Shown here is one of the setups used in our research. Not only is there a motion system controlled by computer software together with nozzles connected to pressure dispensers, but there's also a vision and laser system which allows for real-time detection of objects for a fully automated picking and placing process. With this system, the locations of zebrafish embryos were detected in real time using machine vision, and they were individually picked up out of fresh water. The embryos were then placed on a substrate suitable for submerging in liquid nitrogen, enabling effective organism vitrification. Note that on the angled platform, the nozzle is purged of water residue accumulated during the picking process by using positive pressure. It was found that the pick-and-place system yielded cryopreserved organisms with viability equivalent to manual cryopreservation. The robot also allowed for much higher throughput.